In this class, I am explain about DNA sequencing. Determination of the order of nucleotides of a DNA is called DNA sequencing. Determination of the order of nucleotides of a DNA is called DNA sequencing. It is very important for genetic analysis of DNAs. The nucleotide sequence data is very useful to have a through understanding of traits coded by that DNA. Knowledge of nucleotide sequence data of the DNA is a prerequisite for taking attempts to manipulate the DNA in a desired way. There are three techniques for DNA sequencing: Maxam Gilbert method, Sanger's method, and automated DNA sequencing. First one, Maxam and Gilbert method. Maxam and Gilbert in 1977 developed a chemical degradation method for sequencing the nucleotide of DNAs. In this method, some chemical reagent are used to cut the DNA at specific bases. The DNA is cut into small pieces with the help of a restriction enzyme. Each piece should not be higher than 300 base pair size. They are electrophoresed on a polyacrylamide gel. Individual DNA bands are separated. Each bands contain many identical copies of a DNA. Thus, DNA is isolated from each and every band separately. And DNA isolated from a band is denatured using alkali treatment to separate individual strands of the DNA. Then it is electrophoresed to separate identical single strands of the DNA. As a result of electrophoresis, two DNA bands are formed on the gel. The DNA strands in one band are complementary to DNAs in the other band. These two strands are sequenced by using Maxam Gilbert method separately. And the first strand will give the order of nucleotides in one strand, and the second strand will give the order of nucleotides in its complementary strand. The errors in the sequence data may be corrected by matching the nucleotide sequences of these two DNA strands based on the rules of complementarity between the base in DNA. These sequencing DNA fragment is labeled with P32 phosphate group and subjected in the DNA sequencing procedure. The labeled DNA in the solution is divided into 4 equal parts, each of which is then subjected to a specific chemical treatment to cut the DNA at specific bases. The 4 chemical treatments are as follows. DNA in one tube is treated with dimethyl sulfate and piperidine to cut the DNA at guanine bases. The DMS methylates guanine bases at their N7 position, and the piperidine removes the methylated guanine and sugar residue attached to it. DNA in the second tube is treated with DMS acid and piperidine to cut the DNA at adenine bases. Here, DMS methylates N3 position of adenine. The acid removes the methylated base and piperidine removes the sugar attached to it. DNA in the third tube is treated with hydrazine and piperidine to cut the cytosine or thymine. And the DNA in the fourth tube is treated with hydrazine, NaCl and piperidine to break the DNA at cytosine residues. NaCl enhancing the frequency of cutting at cytosine residues. These four chemically treated DNA solutions are separately loaded in the wells of the polyacrylamide gel and allowed to run across the gel. 7 molar urea is added to the gel to prevent the formation of secondary structures that affects the mobility of the DNA fragments in the gel. Temperature raised by the current is also helpful to prevent the formation of secondary structures. As a result, four parallel series of bands are formed on the gel. The image of radio labeled DNA bands is captured on an X-ray film by means of autoradiography. The autoradiogram thus obtained shows four series of bands which indicate the relative position of respectively nucleotides in the DNA. The base sequence is read from bottom to top. The uppermost band resolves the third nucleotide and the lowermost one resolves the fifth nucleotide. Each and every band identifies a particular nucleotide and its position in the DNA. The preceding base of any base is one base shorter than the previous one. It may occur in the same lane or in any of the other lanes. Therefore, base sequence is read in zigzag pattern. It gives the nucleotide sequence of a sequencing DNA fragments. The complementary strands of the sequencing DNA is then subjected to Maxam Gilbert sequencing to detect its base sequences. The errors in the first sequence is corrected based on complementary matching with the nucleotide sequence of the complementary strand. The missed base may be added to the correct location of the first strand. This is precaution to avoid errors in DNA sequencing. In this way, all fragments of the cell DNA are sequenced and the sequence of these fragments are properly arranged to get the complete base sequence of the DNA. They are arranged in the correct order based on the principle of overlapping fragments. At least a few bases at the end of one DNA fragment are homologous. In some bases at one end of another fragment, such two DNA fragments are brought together as the adjacent fragments. In this way, all these DNA fragments are arranged in a proper order to get the complete sequence of the cell DNA. Thank you.